Okay, so last time we ended up with the story of how the ribosome was guided to the endoplasmic reticulum and how the signal recognition particle combined with the signal peptide would work in concert to deliver that cargo to the ER and begin synthesizing proteins that were destined to be secreted proteins, lysosomal proteins, or transmembrane proteins. And I'll try today to summarize that with a few slides at the beginning of the lecture. But most of what we want to talk about today is tissues. And tissues are a whole new topic. And I should remind you, too, that since we're starting a new topic here today, that there will be an exam in here a week from Wednesday. So it's 5 o'clock, and you'll get on the homepage of BMS 300, you'll get in the next couple of days a list of places where, according to the first letter of your last name, you're meant to go for that exam, which will be from 5 to 6:50 on Wednesday evening. And what we're going to do first, though, is going to talk about tissues and define them. A simple definition of tissues is a group of similar cells performing a similar function. Everything we've talked about so far in here could have been talked about in an amoeba. We haven't really talked very much about cell-cell coordination, and yet the coordination of function between cells is a really important aspect of metazoan biology and physiology. So we'll begin that idea here by talking about the types of tissues. Now, in the late 19th century, the early histologists defined 4 kinds of tissues. They said the 4 kinds of tissues are epithelia, connective tissue, muscle, and nerve. We're going to talk today a little bit about epithelia and connective tissue. Muscle and nerve comes in the next unit, so we're not going to spend much time on that today. But what I want to do today is really kind of contrast the structure of connective tissues with the structure of epithelia. When you think of an epithelium, the first thing to think about an epithelium is first that they form surfaces or boundaries. So one of the ideas we'll try to drive home today is that if you run your tongue along the inside of your cheek, you're actually touching the apical surface, the outside surface of an epithelial series of cells. And that epithelium will be a continuous layer from the inside of your mouth down your esophagus, through your stomach, through your intestine, all the way out through your large intestine and out through the outside world at the other end. And so that's a continuous layer of cells. The cells, the cells are organized into this continuous layer. Sometimes they're a single layer. We'll define that as a simple epithelium. Sometimes they're in several layers. We'll talk about that as a stratified epithelium. In all cases, when you start thinking about or describing an epithelium, one of the first things that come to mind is that the cells are attached to a basal lamina. The basal lamina is a layer of extracellular proteins. Sometimes you'll see the basal lamina referred to as a basement membrane. I don't like to use that terminology because when we talk about membranes in here, we're predominantly talking about membranes as the lipid mosaic model of the membrane, the idea of phospholipids with transmembrane proteins. But the basal lamina, which you'll sometimes see referred to as a basement membrane, is nothing like that. It's extracellular proteins. They are secreted proteins outside the cell. And what we'll see here today is the epithelial cells are attached by special junctions to that basal lamina. One of the important aspects of the basal lamina is that it gives a kind of orientation to the cells. We're going to talk about the apical surface of epithelial cells and the basilar surface of epithelial cells. The basilar surface of the epithelial cells are attached to that extra matrix called the basal lamina. The apical surface face out into the lumen of whatever structure that may be, whether it's the lumen of the kidney tubule or whether it's the cheek inside your mouth. Those are all the idea of the apical surface facing the lumen and the basilar surface being attached to the basal lamina. Another characteristic that I want to emphasize here is that the epithelial cells have the capacity to regenerate. They can replicate themselves. And we know this more or less intuitively. We know that if we cut ourselves, that cut normally will heal. We know that if we take a bite of pizza that's too hot and the melted cheese sticks to the roof of your mouth, it's going to be painful for a while, but you know it'll get fixed. Those cells will replicate themselves in short order, the burn will heal, and you'll be fine. The issue comes, though, that every time these cells divide, there's the unfortunate possibility that they can become a cancer cell. And we're just now beginning to understand all the control mechanisms that prevent cells from becoming cancer cells, or in that unfortunate event, do become cancer cells. So just want to mention in passing here that when you see the term carcinoma, and if you ever look at a pathologist report and see the term carcinoma, that means the cell from which that tumor originated was an epithelial cell. That's the meaning of the term carcinoma, and it brings us back around to the idea that these epithelial cells have the capacity to replicate themselves, but they sometimes make mistakes. But again, if you're going to have a continuous sheet of cells, a continuous layer of cells, then there must be some way in which these cells interact. And so we'll then move on and talk about cell junctions. Now, as we go through the course, we're going to find these cell junctions reappearing in a number of different places, but we'll spell them out initially here in terms of cell junctions with reference to epithelia. The first epithelial cell junction we're going to talk about are the anchoring junctions. And again, as you may recognize, if the cells are oriented on a basal lamina with their basilar surface attached to the extracellular proteins of the basal lamina, there must be anchoring junctions that hold them there. And so we'll talk about the proteins involved in these so-called anchoring junctions, which hold the cells to the underlying basal lamina. We'll see that these are hemidesmosomes. So hemidesmosomes anchor epithelial cells to the underlying basal lamina, but there's a related kind of cell-cell junction called a desmosome. And the desmosome holds cells one to the next. So we'll see how these anchoring junctions function to hold some cells one to the other, and the same cells will be held then to the basal lamina by the hemidesmosomes. Then the second character is going to be the so-called occluding junctions. And the occluding junctions have another name. They're called tight junctions. And the tight junctions then are going to prevent the flow of material between adjacent epithelial cells. And they're going to occlude the space between the neighboring cells. And then finally, there'll be communicating junctions or gap junctions. And the communicating junctions are going to be intercellular junctions that permit cytoplasmic communication from one neighboring cell to the next. And we'll work our way through those. And you had a question.
What does it say between desmosome and hepatosome? What is that little?
Occluding?
No, it says anchoring desmosome. What is that?
This is an ampersand. Try again. Is that better? Okay. So what I want to do now is I want to see if I can call up my slides and walk you through a summary of where we were last time. So the first slide is going to be one that summarizes the movement of the signal peptide, I hope. So what we're looking at here is a summary of much of what we talked about last time in terms of how we guide proteins that have been, whose synthesis has begun on a ribosome, but the proteins that have a signal peptide. So what they're describing here is this is a signal recognition particle. And this red bit here is meant to be the first 17 or 20 amino acids, which represents the signal peptide. Now the signal recognition particle now binds to the signal peptide. Up here we have the small ribosomal subunit, and this heart-shaped structure is the large ribosomal subunit. The blue structure is meant to be the mRNA. We'll be reading from the 5' to the 3' end, and we walk along this, adding amino acids inside the ribosome. But now once the signal recognition particle recognizes that hydrophobic series of amino acids, the signal peptide, this entire machine now moves to the endoplasmic reticulum. When it arrives at the endoplasmic reticulum, the ribosome receptor binds to the ribosome, but more importantly, the signal recognition particle receptor binds the signal recognition particle. Then there's some energy transferred, usually through GTP, and I think it takes 2 to 4 molecules of GTP. But the most important idea here is that what will happen is that when the signal recognition particle receptor lets go of the signal recognition particle, the signal recognition particle goes off into the cytoplasm, and there's an exchange such that this initial series of hydrophobic amino acids is now inserted across this so-called translocon, the transmembrane protein. If you put your phone away, I'd appreciate it. The transmembrane protein in the endoplasmic reticulum. And so now we'll see that for a moment here, this signal peptide is bound to the translocon. And then an enzyme will come along and cut that loose, and we can then thread the rest of the membrane, the rest of the protein across the translocon. So this is where we're left with the last one. There's another enzyme here. It clips loose that signal peptide, and the rest of the protein now is fed through the translocon and ends up here in the lumen of the ER. This could be either a secreted protein or it could be a lysosomal protein. Once inside the ER, these proteins will be glycosylated, and then they're beginning their travel from the endoplasmic reticulum to the Golgi apparatus to the vesicles to either the lysosomes or the plasma membrane. But the question remains from this is, how do we manage to get a transmembrane protein? And that they're trying to show here. So we have eliminated all of the rest of the illustration showing the ribosome. But recognize that the ribosome with the signal recognition particle and all of that will have delivered this thing to the translocon. And it will have threaded this initial series of signal peptide across the translocon. But then, as protein synthesis continues, we encounter this region up here in orange, which is a stop transfer sequence. When that happens, because it's hydrophobic, it sticks to the hydrophobic amino acids in the translocon. It kind of gets hung up here. Then, just as we saw in the last image, there's a signal peptidase, clips off the signal peptide, but now this translocon opens up like a clamshell and deposits this transmembrane region, this so-called stop transfer sequence, in the phospholipid bilayer. So this is going to be a series of 60 or 70 predominantly hydrophobic amino acids will be deposited across the membrane. This can occur, now this is showing what happens for a single transmembrane protein, but we can do this multiple times. So in this case, there are these internal, what they call start and stop transfer sequences. They too, because they're hydrophobic, at least initially will bind to a signal recognition particle. The signal recognition particle then will hand off this original start transfer sequence to the translocon. Then synthesis will continue onward until we reach the stop transfer sequence, and the translocon opens up, and now we deposit both of these transmembrane regions across the phospholipid bilayer. And this will happen, this may continue to have another 6 or 8 additional, maybe more, transmembrane protein regions, which will be hydrophobic, will run into these start and stop transfer sequences that will bind to the hydrophobic amino acids in the translocon, and they will open up and subsequently make a multi-pass transmembrane protein. All of those regions that go across the phospholipid bilayer will be hydrophobic. So any questions on that? That pretty much summarizes where we stopped last time. So understand the ideas about the signal peptide, understand the importance of the signal recognition particle, understand the hydrophobic nature of those amino acids and their ability to bind to the translocon. If you keep all of that in mind and can recognize that that's the mechanism we use for generating those transmembrane proteins and secreted proteins generated on rough ER that we talked about a couple of weeks ago. So any questions on that then? Not, let's start into tissues. And of course, we'll start here with the definition. And the definition is a group of similar cells working together to carry out a similar function. Now, as I suggested in the introduction, up to now, everything we've focused on has been about individual cells, single cells. And we could have been talking about any eukaryotic cell on the planet. But now we're beginning to think about how cells work in concert, how they're organized to work together to perform the functions that we know in the human body. And so what I want to do here is simply lay out this idea of types of tissues. And the types of tissues were described in the 19th century. And the 19th century histologists said all of the human body can be divided into 4 tissue types. There will be epithelia. There will be connective tissue. There will be muscle. And there'll be nervous tissue. We have several lectures dedicated to muscle and nerve, so we're going to leave these behind at this point. But I do want to spend a little bit of time on the generalities of these 2 tissue types and try to give you some comparing and contrasting of these 2 tissue types. When we talk about epithelia, we think about a continuous layer of cells that sit on a basal lamina of extracellular protein. So the image that I like to hold in my head when I think about that is that there are these blue boxes which we can refer to as epithelial cells. These cells are stacked shoulder to shoulder. But again, I don't do justice to this in these 2 dimensions because they are going to come out from the plane of the blackboard and go behind the plane of the blackboard. They're a great sheet, a great continuous sheet of epithelial cells. All of the epithelial cells sit on an extracellular matrix. These are extracellular proteins. This is an extracellular matrix of proteins, and this has a name called the basal lamina. Now, one of the hallmarks of all of these epithelia is the epithelia. Epithelium is singular, epithelia is plural. Epithelia form boundaries or surfaces. In most cases, these epithelia, to be a true epithelium, will extend to the outside world. So again, as I said in the introduction, you can imagine the epithelium forming the lining of your cheek. Now, it's not quite some mucus over that, and in the inside of your mouth, it's probably not a simple layer of epithelial cells. But nonetheless, there will be a continuous layer of epithelial cells from the inside of your mouth, on your tongue, down your throat, through your esophagus, all the way down to your stomach, throughout the gastrointestinal tract. There will be a continuous layer of epithelia, and it will be continuous from your mouth to your anus, and so it's continuous to the outside world. When we start talking about the respiratory system, we're going to talk about a respiratory epithelium, and there will be epithelial cells in your lung that will be exposed to the air. Those epithelial cells, you can stand on those epithelial cells and walk from one epithelial cell to the next, from the lung, out through the bronchioles, out through the trachea, out to the pharynx, to the outside world. So it's a continuous layer of epithelial cells. When we get to the kidneys, when we talk about the kidney epithelium, we'll find that there are cells in the kidney that form as the nephron. We can walk from one epithelial cell in the nephron to the next, and going from the nephron to the collecting duct to the ureter to the bladder to the urethra to the outside world, a continuous layer of epithelial cells. And we'll see this pattern over and over again, where we have a continuous layer of epithelial cells, and these epithelial cells sit on a basal lamina. So hold on to the idea that they're a continuous layer of cells. For the most part, we'll talk about a single layer. And the single layer is called a simple epithelium. In some cases, particularly when we get to skin, which is another classic epithelium, the cells will be stacked, and this is a stratified epithelium. But even in the case where you have several layers of the epithelial cells, at least one layer needs to be anchored, needs to be attached onto this basal lamina. The other important point to make here is that because one surface of the epithelial cells sits on a basal lamina, it gives us a kind of orientation for the cells, such that this surface of the cell, this region of the plasma membrane that's attached to the basal lamina, is called the basilar surface. And then the other side of the cell, up here, which is facing the lumen of the tube, is the apical, the apex, the top, the peak. And so the apical surface is the surface of the epithelium that you can touch with your tongue. That epithelial surface then, this apical surface, is now open to the lumen of your mouth. And if you were in the stomach, it'll be open to the lumen of the stomach. If you were in the small intestine, it'll be open to the lumen of the small intestine. Lumen simply meaning the inside of the tube. Just as we talked about the lumen of the endoplasmic reticulum, but the inside of the endoplasmic reticulum. But here we have tubes that go to the outside world, and they are lined with an epithelium. Before we get down into the weeds of some of the other details of these epithelial cells, let me mention here what we mean by connective tissue. Connective tissue is divided into 4 connective tissue types. The first connective tissue type is said to be connective tissue proper. Now, connective tissue proper is one that we're all more or less familiar with. The connective tissue proper includes things like ligaments and tendons and the dermis of skin. It's particularly characterized by collagen and elastin. Collagen and elastin are extracellular proteins. Secreted by connective tissue cells. In fact, what we're going to see as we walk our way through the different kinds of connective tissue is that this is a hallmark of all connective tissue types. The connective tissue types, whether we're going to talk about connective tissue proper, or whether we talk about cartilage, or whether we talk about bone, or the most peculiar of them all, blood. All of these are characterized by scattered cells embedded in a protein matrix they secrete. Now, the cell type that's going to be secreting the collagen and the elastin for connective tissue proper, that cell type is said to be a fibrocyte. Sometimes you'll see the terminology that rather than saying a fibrocyte, it'll be termed a fibroblast. And the term blast simply means an incompletely differentiated cell. But a fibrocyte is a completely differentiated cell among the connective tissue that secretes extracellular proteins, mostly collagen and elastin. But that same pattern is also true for cartilage. Cartilage comes, I'll go back to the upper panel and spell this out. Cartilage comes in 4 types. So cartilage is a connective tissue. But again, if we look at this, we see there are these cells. And I'll show this in blue. And they're separated one from another. So these are meant to be chondrocytes. And the theme here of connective tissue is widely scattered cells embedded in an extracellular protein matrix that they secrete. So these chondrocytes, their job is to secrete these extracellular proteins. So they'll have very busy Rough-ER and Golgi producing these extracellular proteins. The ones I'm putting in here in red are typically collagen. But in addition to the collagen, there are these other proteins that have this spring-like structure. And these spring-like proteins have a name called elastin. And as the name indicates, these elastin proteins are stretchy, so that when you tug on some of these cartilage structures, they're actually a little bit stretchy because of the elastin, yes?
So how do those differ from the fibrocytes? Like how do chondrocytes?
So the question here is, and we can tell the story in all cases. The question here is, you've just told me the connective tissue proper is laid down by fibrocytes, and the major proteins that the fibrocytes secrete are collagen and elastin. And then I've told you that cartilage is also a connective tissue, and it's also widely scattered cells that are secreting collagen and elastin. The distinction here is in part where these cells came from. The cartilage cells, the cells that are going to produce cartilage, the chondrocytes, have a cell origin in something called chondroblasts. And these guys start out as fibroblasts. In all cases, these connective tissue types originate from something called mesenchyme. So embryologically, they have separate cell types from which they originate. Now, there's a common stem cell for all of these, but those common stem cells will differentiate into the ability to be connective tissue proper, cartilage, bone, or blood. Now, having said that, one of the major distinctions between even something like a ligament and cartilage is going to be the proportion of these extracellular proteins that they secrete. They just don't secrete collagen and elastin. They secrete other proteins as well. But in large measure, it's the proportion of these extracellular proteins that these cells are secreting that will determine their function and their name. And I can draw that out by talking about cartilage itself. Because if we talk about cartilage comes also as 4 separate types. There's hyaline, there's fibrocartilage, actually there are only 3, and elastic cartilage. Now, all of these have this general characteristic of having these chondrocytes separated one from another. But what distinguishes these kinds of cartilage one from the other is the relative proportion of proteins that they've secreted. Now, hyaline cartilage includes a lot of something called Glycosaminoglycan. The way I like to describe glycosaminoglycan is this extracellular stuff. It's this extracellular protein. Let's call this as a glycosaminoglycan. Now, any of you who have worked with fiberglass will recognize this pattern. In fiberglass, what you do is you take these filaments of spun glass. You put down a layer of these filaments of spun glass, and then you smooth into that layer of spun glass some sort of resin, usually an epoxy resin. And then you lay down another layer of spun glass, fiberglass, and then on top of that, you lay down another layer of resin. Well, the analogy there is that these collagen and elastin filaments are extracellular proteins akin to the fiberglass filaments, the glass filaments. And then the glycosaminoglycan is equivalent to the epoxy, which you then spread in and give it strength. So we do the same thing here. We secrete the glycosaminoglycans. These are highly negatively charged glycoproteins. They are secreted out among the filamentous-like collagen and more elastic elastins, and they form a fairly hard hyaline cartilage. You know about hyaline cartilage. If you've ever eaten a drumstick, you know about hyaline cartilage, because when you break the drumstick free of the thigh, there's hyaline cartilage covering that joint surface. That's a kind of cartilage. It's made mostly of collagen and the glycosaminoglycans. The fibrocartilage, again, is going to be rich in collagen. Where we find fibrocartilage is predominantly between the vertebrae and the spinal column. That's fibrocartilage. Another place where we find fibrocartilage is the meniscus. Of the knee. These fibrocartilages tend to be rich in collagen because they're tough. So you need this tough rope-like structure to form the pads in between your vertebrae, because every time you turn, every time you twist, you put a lot of torque on those. And it's also true for the meniscus of the knee. And commonly we tear these. Most soccer players know about a torn meniscus. And so what will happen is in the knee, this cushioning fibrocartilage will often be torn. It's tough, but it's not impervious. And then finally, the one I think that's the easiest to recognize is elastic cartilage, and this is found in the epiglottis and most commonly in the outer ear, the pinna of the ear. The external ear is really just skin, epithelium, draped over cartilage. But it's not like the meniscus, which is really tough. It's pretty stretchy, and we know you can take your outer ear and fold it over, and thank God it returns to its original shape. And so that's largely due to the fact there's so much elastin out here. So the elastic cartilages are enriched for elastin. And again, they have this spring-like structure. So when we pull on them, they can get longer. But they also have this ability to recoil so they can return to their original shape. That gives them their elastic characteristics. We'll run into this elastic cartilage, we'll run into this form of extracellular protein when we talk about the dermis of the skin, because it too is enriched for elastin. And that's the reason why you can pinch your skin and it comes back to its original shape. And as over time, these elastin molecules break down in the skin, they lose their resilience and wrinkles form. And so this is the character which, although it provides the elasticity of skin, as we age, that elasticity breaks down. But again, the major idea here is when you think about a connective tissue, hold on to the idea that we have widely scattered cells embedded in an extracellular matrix. There is a fourth one here, which I missed up above. Oh, it's bone, right. I have it here. So bone also is connective tissue. But bone as a connective tissue secretes, let me give a cell type. These will be the osteocytes. And the osteocytes secrete collagen, mostly. But they also secrete something else called hydroxyapatite. Hydroxyapatite is basically calcium phosphate. With some magnesium phosphate mixed in. This is what makes bone hard. This is what makes bone dense. So what distinguishes bone from the other connective tissues is the fact that it contains widely scattered cells, but these cells, these osteocytes, secrete, in addition to proteins, they also secrete a mineral, calcium phosphate. And then finally, blood. Blood originates from cells called hematopoietic stem cells. Hematopoietic stem cells give rise to all of the cells of blood. These hematopoietic stem cells reside in adults, mostly, almost entirely, in the red bone marrow. So the hematopoietic stem cells, hematopoiesis is the process of making blood cells. And the hematopoietic stem cells, let me spell it out. Hematopoietic stem cells give rise to erythrocytes. They give rise to cells of the immune system. And that was the final piece here. Recognize that these tissues, the blood, in fact, obeys the same rules that we talked about for the chondrocytes or the fibrocytes or the osteocytes. And that is that these cells, whether they're erythrocytes or cells of the immune system, are embedded in an extracellular extracellular protein matrix, which is in fact the protein inside blood, the protein of blood. In fact, those proteins will coagulate and form a clot through a whole cascade of events. So those are extracellular proteins, and these cells are embedded in those extracellular proteins. So the thread that runs through all of these connective tissue types is cells separate one from the other, but embedded in a protein matrix that they have secreted. And that protein matrix may be as arcane as the proteins in blood, or they may be as solid as the collagen and elastic filaments that you see in the kinds of connective tissue proper and in cartilage. So any questions on that? It's basically all I want to say about connective tissue, and we'll come back to bone a little later. But the ideas that I want to finish here are mostly to do with the epithelium. So that we've described the epithelium as a layer of cells, a continuous layer of cells, cells stacked one next to the other, all of whom are attached to the basal lamina or attached to neighboring cells, but they form a continuous layer of cells. The other characteristic of these epithelial cells, let me spell out the characteristics. So the characteristics of epithelial cells, of an epithelium. We've described first that they're a continuous layer. Secondly, they are attached to a basal lamina. The third issue about these cells that we haven't quite described yet is that they replicate. They divide to replace themselves. We know this. Your skin, the epidermis of your skin, we'll talk about this in more detail, but the outer layer of your skin is an epithelium. And we know that if you cut that skin, you know that within a few days it will heal. It will heal because these epithelial cells replicate. You know if you burn the surface of your mouth, it will heal in a couple of days. And it heals because these cells divide and replace themselves. All along your GI tract, you have cells that are replacing them. You have epithelial cells that are replacing themselves every minute of every day. And in fact, one of the real problems with radiation poisoning, what will commonly kill people with radiation poisoning, is the radiation is high enough. When the radiation doses are high enough, they will kill the dividing cells all along the intestine. Then the intestinal epithelium breaks down, and people break down from the destruction of that epithelial barrier. And so, again, just pointing out that these epithelial cells must divide and replace themselves. And they normally do this just fine. But the other problem here is that, on occasion, epithelial cells give rise or generate a tumor of cancer cells. The issue appears to be that every time a cell divides, it has this small but unfortunately finite probability of making a mistake that it loses control of its cell division machinery. And when it loses its control of its cell division machinery, it begins to divide out of control. And when this happens, this forms something known as a carcinoma. We're beginning to understand a lot more about the biology of these carcinoma cells. And one of the issues that's been known for quite some time now is that when an epithelial cell becomes a cancer cell, one of its problems, one of the reasons it becomes dangerous, is what happens is they say it loses its respect for the basal lamina. It no longer properly attaches to the basal lamina. Because it's no longer properly attached to the basal lamina, in combination with this inability to control cell division, these cells now leave their connection to the basal lamina and wander off someplace else. They may wander off to the bone, they may wander off to the liver, they may wander off to the brain, and then they continue dividing. But because they're no longer attached to the basal lamina, they then form a tumor. And when you start getting these cells wandering very far from the initial site of the tumor, you have metastatic disease. And the greater the metastatic disease, typically the worse the prognosis, because it's difficult to control these cells out of controlled division in a variety of different places. But we're now beginning to recognize that there are a whole host of control mechanisms involved with the anchoring junctions we'll talk about here in a little bit, that anchor these epithelial cells to the basal lamina. It appears that there are a whole host of proteins that attach these cells to the basal lamina, and they include control systems that say, you must stay attached to the basal lamina. And when those control systems get perturbed, then these cells no longer stick to the basal lamina and go off and try to live someplace else. But one of the major treatments right now for cancer is to try to, through surgery, excise those tumors and hope they have these rogue cells have not gone too far. But it all comes back to the epithelial cells and their attachment to the basal lamina. Yes. I'm hearing voices, yes. So the question here is melanoma a form of carcinoma? And the melanoma is a little different. And the reason for that is that the origin of the melanocytes, which give rise to the melanomas, don't come strictly from an epithelium. They originate as a series of cells called neural crest cells. Now it's true those are original ectoderm in development, but during development there are a series of cells along what is known as the neural tube. As the nervous system begins to generate, there are these cells called neural crest cells, and they start to wander off into the periphery. And they make a whole bunch of things. They make the neurons in the sympathetic ganglia. They make the cells, the sensory neurons in the dorsal root ganglia. And we'll talk about both of those later on. And they also include the Schwann cells, which are glial cells in the periphery. And they also include the cell type, which is the cell that you're talking about that generates the melanocyte. So those all come from that origin. So they are given a different name. But having said that, because they work their way back to an original ectodermal origin, there's some indication that some treatments for these melanomas can be used that also treat nervous system problems. And the hint here is that there are some treatments that are using herpes viruses to deliver certain characteristics to these melanocytes, these malignant melanocytes, and they'll actually deliver the herpes virus to help kill that melanocyte, and then they'll introduce a whole immune response to those melanocytes, so your immune system will see it. But that has to do with the fact that they're related all the way back embryologically to the ectoderm and those ectodermal cells. But they're not considered, as far as I know, to be a true epithelium. Let's leave it at that.